everyone, welcome to Science Friday.
Here's some for E as well. The one on the top here is one of the ways that you can define E.
What makes a field more likely to have an unbalanced gender distribution?
So this can be, like, in the case of this paper, gyrification or brain folds.
A neuroscientist started to look at the brain
as like the
more as an
interconnected.
I'm asking incredibly negative aspects
Not
to
to
what
define. They're terrifying.
So first I want to, you hear this term precision medicine, and I want to define that in a little bit. But first I want to talk a little bit about treatment and how big of an impact it has on people's lives and those around them. So I want to start off with an example of a person I know, someone with Parkinson's disease. So for those of you who know about Parkinson's, this is a condition that is characterized by a lot of symptoms that get progressively worse over time and have a big impact on people's day-to-day lives. So a couple of examples of symptoms include slow thinking, tremors in arms and legs, shuffling movement, and because of these very apparent symptoms, patients really need treatment right in diagnosis as soon as these symptoms start becoming apparent. And with this patient, their doctor initially recommended a certain medication for them to take, which they took for a long period of time, 5 years. And over the course of those years, they became increasingly tolerant to the effects of the drug, and their symptoms began showing. So towards the end, in the last year or two, they had to transition and start adding more doses of this drug, start increasing the amount in order for it to have the same effectiveness it had at the beginning. But with an increased dosage of a drug that already has side effects, of course those side effects will build up and increase. And so there comes now this tension between the side effects of a drug and its effect that you have to continually increase the dose to arrive at the same sort of efficacy. And that's what happened for the next 6-month period after this increased dosage. When these side effects became apparent, the patient had to scale down on the first drug, kind of add in a second drug, test out different combinations of those, a lot of trial and error, and there was this constant fluctuation between symptoms and side effects. And of course, over time, this balance grew finer, but it was something that was affecting their lives every day. And this isn't just a one-off problem. Overall, with the variety of medications available nowadays, people tend to undergo more treatment that's less effective for an extended period of time. And you can imagine for patients with diseases like Parkinson's, how big of an effect that has on their lives. And so we have so much technology available nowadays that can glean information about all aspects of our lives. But in particular, the National Institutes of Health determined 3 broad categories that they define as our determinants of health. And these are very general, and in total, they play important roles in determining how each of us can react to different treatments. So I want to open it up to you guys. What are some guesses for very broad categories that may play a role in determining our health? Mental, think even broader, like very, very, very broad. So I'll give a hint. For the first one, think about what the kind of most distilled and the most scaled down unit of each of us that makes us unique from every other human being. What is it we learn about in biology that we know is the most basic unit of each of our bodies? DNA. So the first determinant of health is our genetics. Obviously, every person is genetically different from every other person, and so this plays a large role in determining how we react to treatments. For the second one, think about the world around us, perhaps like things like weather, things like where you live, like pollution, things you can't control. What category might those fall under? Environment. And the last one is more related to our choices, the way we choose to live our lives, so things like exercise and nutrition. What category may that fall under? Behavior. Pretty much. Lifestyle. Just another word. And these 3 categories in total can be used to really determine how a patient might react to a treatment as opposed to every other person in the world. And you can imagine each of these are different for every single person. And this gets to the concept of precision medicine, which is a holistic approach to treatment that takes into account all three of these factors to tailor a person's treatment to them from the very beginning, from the onset of their diagnosis. Meaning there's no trial and error period, there's no fluctuation, or at least that period is minimized for patients. And so we get to the question of what does that really mean in healthcare? Well, with the technology that I mentioned before, we can kind of measure a couple of different levels of information from our bodies. And this is called multi-omics, as you'll see in a minute, because these levels each end in this suffix of omics. Starting with the very lowest and most basic level of genomics, looking at our DNA and how that differs between people. Moving on to RNA, which is the next step in the central dogma for those of you who have taken biology, how our differing DNA and also environmental situations affect how that's transcribed in our RNA, and then into the last step of proteomics, which is our protein expression and function. And then finally, thinking about how all of these play a role in the various bodily processes that go on, which is known as metabolism, and the final products, which are metabolites. So these are 4 different levels of information we can glean from every single person, and that are different for every individual. And this is just a picture that depicts that sort of central dogma concept and how each of these levels are tied together inherently to affect our body and our individual characteristics. So the lab I worked at over the summer focused on a specific subset of diseases known as inflammatory bowel disease. We're gonna kind of narrow that down further to one study that focused on ulcerative colitis, which is a condition that's mostly localized in the colon, which you can see in kind of brown color here surrounding the intestines. And in short, there is inflammation and ulcers, which are also sores, that patients get in their colon that cause them to have issues in their digestive tract. And a common treatment for ulcerative colitis is vedolizumab, a drug known as VDZ. So basically how this works is that in ulcerative colitis, you have these white blood cells known as lymphocytes that cross from the blood vessel into the colon membrane. Once they do that, they begin recruiting pro-inflammatory markers that cause inflammation around the colon, and that leads to the inflammation and ulcers I was talking about before. So vedolizumab is this purple sort of triangle or 3-pronged shaped protein that binds to a cell surface protein known as alpha-4 beta-7 on these white blood cells. Essentially, all this green protein does is it allows these white blood cells to cross from the blood vessel into the membrane, and by binding to it, vedolizumab prevents the white blood cells from then crossing into the colon and causing inflammation. And this is a very effective treatment for numerous patients, but there is a question around how effective it is for patients for whom this treatment doesn't seem to work as well. So there's 2 kind of different groups here, responders and non-responders. Patients for whom this treatment is very effective, and patients for whom, sort of mysteriously, the treatment is not effective for. And we just discussed this mechanism of ulcerative colitis and how vedolizumab completely inhibits it. And so what could it be that might be causing continued inflammation and ulcers in certain patients who are treated with this drug? And to answer that question, the researchers developed what they called a streamlined multi-omics pipeline. Multi-omics, the kind of different levels of information about our bodies that we discussed before, and then streamlined, meaning that making this in a way that can be feasibly implemented in a healthcare setting. So making this multi-omics analysis cost efficient, time efficient. This, in tandem with answering the question on the previous slide, were the 2 goals of the researchers. So to explore these questions, the researchers gathered 3 groups of patients, patients who were healthy controls, patients with ulcerative colitis, and then patients with ulcerative colitis who were treated with vedolizumab. The researchers first performed a series of single-cell analyses on these patients to look at cell-by-cell differences between each of these patients, and in particular between groups. And then after determining this kind of cell-by-cell differences, the researchers took a step back and did spatial analysis, which is looking into how these differences actually manifest in the way these cells interact with each other and with the tissue itself. So starting off with a single cell analysis, there were many cell types that were actually affected by vedolizumab. A lot of them through indirect mechanisms, coming from the one we described before, but 3 in particular were really interesting because of the way they tied together and this kind of cohesive story that they created. So the first one, and just to kind of explain these graphs really briefly, the y-axis is cell frequency, or the number of cells in patients. And each of these bars represents a different group, with the gray bar being healthy patients, red bar being diseased patients, and a blue bar being treated diseased patients treated with vedolizumab. So in the first graph, which is looking at mononuclear phagocytes, these are cells that kill bacteria and cause inflammation in the body. You may know them as macrophages. We see a downward trend in the number of cells in patients treated with vedolizumab when compared to patients who are just diseased and untreated. And these cells, as I mentioned before, play a role in inflammation in the body. So reduced number of cells indicates less inflammation going on following treatment. A similar pattern is displayed with stromal activated fibroblasts. This cell type is a bit more interesting because typically in the body, fibroblasts actually play a role of healing scar tissue. So it's usually determined as more of a healing marker, but as I mentioned before, the researchers did multi-omics analysis on this in this study. And one of the benefits of that is understanding really all angles of each of the mechanisms that they discover. And so in addition to looking at the number of cells that were present in the patients, the researchers also performed genetic analysis on these cells to determine what really is their role specifically within the context of this disease. And with these fibroblasts, they found that they actually recruited more inflammatory markers than the healing markers that are typically associated with the cell type. So even though it's a bit counterintuitive, in this setting, reduced activated fibroblasts actually indicates less inflammation, just like mononuclear phagocytes. And then finally, the last graph is looking at a third cell type known as epithelial cells. These you can think of as the membrane cells. These make up the membrane of the colon. And there is an inverse trend here where there is an increase in epithelial cells following treatment. What this indicates is that the patients who have now taken the drug and have reduced inflammation in their colon are now able to heal their membrane, and the epithelial cell counts are growing as that membrane is healing. So with this data, the researchers proposed a model whereby vedolizumab, the drug, reduces inflammatory mononuclear phagocytes and fibroblasts, which cause an increase in epithelial cells that indicate mucosal healing. And mucosal is just another name for membrane. So basically what I just explained. And then the researchers wanted to move on to the spatial analysis. Now that they determined these cell-by-cell changes, they want to see how does that affect the tissue and how these cells interact with each other. So each row of these 2 figures represents a different form of spatial analysis the researchers did. Also, I'm sort of vastly distilling all of the different tests and analyses the researchers ran in this study, in this presentation. These were mainly just the figures that were kind of more visually indicative of the data the researchers gathered, but there was a whole host of different cells and different contexts that the researchers actually measured in the study. So there's a ton of data that they were able to gather. But this data is in particular relevant to their main findings. So in these panels on the left, these are each kind of images of colon tissue in the different groups. The left panel is the healthy patients, middle is ulcerative colitis patients who have been who are untreated, and then all the way to the right is patients who are treated with vedolizumab. The different dots represent different cell types, blue for mononuclear phagocytes, yellow for fibroblasts. These are the 2 inflammatory cells that I mentioned before. And so, starting with healthy controls, you can see that the cells are much more dispersed than they are in the diseased patients, where there's many more cells and they're much closer in proximity. But looking at the vedolizumab patients, the ones who had the disease and then were treated with the drugs, we see an inverse trend, where the patient's cell counts are now trending back towards how they were in healthy controls, where they're more dispersed and less clumped together. So what this means is that here in diseased patients, these cell types are interfacing very closely, and they're recruiting each other to cause inflammation in the colon. So this is, as I showed the image in a couple slides ago, where the colon is really inflamed and has a lot of ulcers happening because of the presence of these cells. With vedolizumab in patients, these cells are now being reduced because they're being prevented from crossing into the colon membrane, and there's less quantity of them that are closer together in proximity. The researchers quantitatively describe this in these box and whisker plots to the right. with the different colors representing the different control groups or the different groups that I described before. And then on the y-axis is just the distance between cells in each of these panels. And you can see here that the blue box in both of those graphs has a higher distance between cells than the red box, which is these diseased patient panels. So with this information, the researchers now extended on the model in the previous slide by claiming that mononuclear phagocytes and inflammatory fibroblasts, these yellow and blue cells, are increased in density and proximity in ulcerative colitis tissue, and this pattern is inhibited by vedolizumab. So this may seem obvious, but it's novel in the sense that historically, vedolizumab has been really explored through its effect on lymphocytes, which are the white blood cells I was describing. But its ability to actually directly affect these inflammatory cells is particularly significant. And with this information, the researchers then kind of geared back towards that central question that I was describing at the beginning, determining what can we actually find out about patients before they are treated to see if this treatment will be effective for them or not. And this gets back to the determinants of health I was describing before. How do we know whether a treatment might work for one patient and not work for a completely different patient? So in these graphs on the left, on the y-axis is a bunch of different cell types that the researchers looked at. The x-axis is something called a net enrichment score. For the purposes of this presentation, it's not really important what it is or how it's calculated. What matters is understanding that here the negative values actually don't mean anything. Really what's important is the magnitude of each of these bars. So how far away a bar is from the center line indicates the number of cells in that patient either before or after their treatment. So in looking at responders, first of all, before treatment, they had high levels of all of these different types of cells. In general, this indicates the amount of inflammation that's going on in their colon. And then post-treatment, all of those zero out. But there's an inverse trend with these epithelial cells that I mentioned before. And if you remember, these are the membrane cells that indicate healing going on. So this is really confirming the findings that the researchers found in their study. I should mention, this is actually data that's determined from large publicly available data sets online. So the researchers wanted to combine this existing data with their data to kind of make predictions about this disease and come to conclusions. So this publicly available data is now confirming what the researchers found. And then looking at non-responders, we see a much less clear-cut picture. So the gray bars here are cell counts that were not highly significantly expressed enough to actually be important to make conclusions on. And so we'll focus on the red bars, which you can see are further away from the middle line, indicating that there were high enough numbers of cell counts. And in non-responders, we see before treatment, there was a very high level of cytotoxic lymphocytes. These are white blood cells that are toxic to other cells, kill the cells around them. And then following treatment, the patients actually have a continued level of inflammation going on, characterized by the high level of activated fibroblasts and stromal cells that are shown here. And this is very interesting because these patients, having taken vedolizumab, which really, as we described before, works to inhibit inflammation by preventing the white blood cells from going into the colon membrane, having taken that drug, the patients are still having inflammation. And so it's not as effective. And then this leads to an insignificant, well, in here there's an insignificant level of epithelial cells, showing that there's not as much healing going on in non-responders as there is in responders. And I'll get to that in a little bit, but first I want to describe this graph all the way to the right. And this one is really getting to the heart of that question of how can we figure out whether a patient will be a non-responder or a responder before going through that whole trial and error period that takes so much time. And so the researchers looked to quantify certain cell types in patients who were non-responders that were not present in patients who were responders. Meaning that the researchers want to see, can we run tests on patients and see what their cell compositions are, and then from that data, determine whether this drug will be effective or not effective. And there are a number of cell types that were highly expressed in non-responders. We won't go into too much depth on what the role of those cells are, but what's important to understand is that this kind of analysis can actually play a big role in the sort of predictive precision medicine modeling that could be important in healthcare. And with this data and with the data they previously gathered, the researchers came to a final conclusion that vedolizumab non-responders have higher pre-treatment tissue inflammation, as we talked here, driven by alpha-4 beta-7 independent pathways. And this is getting back to this kind of data here, where there's continued inflammation going on in patients who've taken this drug. As we talked about before, vedolizumab works through this alpha-4 beta-7 protein that it binds to and prevents white blood cells from growing into the colon. By having continued inflammation following treatment with the drug, this indicates that certain patients have inflammation going on that is not just caused by this alpha-4 beta-7 protein, but is caused by other mechanisms. And that's what the researchers mean by alpha-4 beta-7 independent pathways. What's really interesting is the fact that for some people, alpha-4 beta-7 was the primary mechanism of inflammation. This drug worked. It was very effective. For others, it was not, and the drug was not as effective. And this is the kind of reasoning that precision medicine is really working towards. And so now we get to the sort of broader scope. What does this mean really for our healthcare system? The researchers did this analysis for a certain drug. They found conclusions that could play a role in determining predictive algorithms that they could use when thinking about whether patients will be responders or non-responders. But that's not actually what's going on today in healthcare as it exists. Currently, physicians are approaching patients more as a one-size-fits-all model, meaning that a patient who has a condition, but might be in a completely different situation from another patient who has the same condition, would often receive a similar medication and a similar treatment timeline. And so that gets back to this whole trial and error period where each patient is then fine-tuning their own medication schedule. Now, with precision medicine and by taking into consideration those determinants of health that I described earlier, we can move towards a model of healthcare that's approaching each patient more as an N of one, or as a sample size of one. What this means is that from the onset, physicians rather than patients are the ones who are really taking into consideration those patients' genetics, their environment, their lifestyle, and thinking about how effective a treatment might be for them based on those determinants. So with all the things, there are challenges, starting with high costs. The analysis run in this study was very expensive, but remember that streamlined aspect of the multi-omics pipeline I mentioned at the beginning. Part of that goal is making this less expensive and more time efficient and cost efficient. So this is work that's being actively done, and costs are gradually lowering. Also important to mention that with precision medicine, the long-term benefits of might be better to give an example. So a patient comes in who receives a treatment that's a typical treatment for their condition. In the future, they come back, they have more visits with their doctor where they check in about how well the treatment's working, they get new prescriptions, they change dosages. All of those reduce costs in the healthcare system. With precision medicine, the goal is that from the beginning, the patient would have a medication that's maybe not tailored perfectly, but more optimized to their actual condition. And so they wouldn't have to go with those constant check-ins and those new prescriptions, all of those that induce additional costs in our healthcare system. With the problem of, with the situation of gathering so much data that is defined, that multi-omics is defined by, there is obviously the question of data security. Maintaining all this data in a secure environment, and then getting to this issue of ethics. So this is a very interesting point that I could delve on further, but I'll just briefly mention. Things like having now information on a patient's genetics and their environment and lifestyle. How might insurance companies revise their policies to then group different patients in different, whether patients will be able to receive insurance for certain treatments or not, just based on maybe genetic differences or where they live, And then incidental findings such as a 20-year-old coming to the clinic having a routine genetic test run on them, and their physician finding out they may have a life-threatening disease that could immobilize them in 20 years. Is that something that is ethical to tell a patient when they're at full capacity in the age of 20? That's the kind of question that will come up more often in precision medicine, where there's so much more data that's available. Having treating each patient as an individual requires a large data set to be able to see, okay, how do patients differ between populations and between different groups? We need to have data that is representative across many populations. And this leads to the question of health disparities or health inequalities. In our research nowadays, and not just in access to healthcare, but in research, there is a disproportionate amount of data that's gathered on certain populations like white people than minorities, such as blacks or Hispanics. This is a problem now. It's not just a potential barrier with precision medicine, it's a barrier to our ability to provide good treatments to patients in the present. But this will become accentuated if we base our treatment schedules and regimens even more on the data we have available. And this is something that needs to be addressed as we move into a world of precision medicine. And we are moving into a world of precision medicine. Through oncology, psychology, cardiology, there are so many applications that are becoming apparent in the modern world. And it's something that's happening now. By 2030 maybe, that's a big claim, but we could be getting into a precision medicine world very soon. And this is further evidenced by the White House in 2015 announcing precision medicine as one of the priorities for the National Institutes of Health in the coming decades. And this all gets back to the point about quality of life and what healthcare really means. What's the ultimate goal of our healthcare? It's to improve the lives of our patients. And with precision medicine, we're moving more towards a model of healthcare where we're improving the ability of patients to live their lives and to have better experience with their treatment regimens. So I want to acknowledge my amazing lab, my PI, Mike Kataw, for his endless patience and kindness in describing so many of these concepts. Elvira and Kathy for their continual mentorship over the summer. I really could not have learned so much without any of them. And before I conclude, I just want to briefly mention that this is the paper that I referenced. Notice how the first 2 lines of the citation consist of authors with an ellipses, cutting out even more authors in the middle. This is how much collaboration is required in the world that is to come, in the world of precision medicine. And we're really entering a model of healthcare where we're needed to work together in order to do the best for our patients and improve the world. So thank you all for listening, and I want to open it up to questions.